All right. So we're going to transport ourselves back to the 1960s. And this was right after, I don't have time to go into all this, but right after kind of the golden era of DNA when we discovered the structure of DNA, this double helix with these bases, and we're unraveling that mystery about the structure of DNA. And now we're interested in, what makes a stem cell a stem cell? What is it about this powerful cell that can then become all other cells? And there was a huge question at the time that was answered by this gentleman called John Gurdon. His name is John Gurdon. And he studied the same little frog that I did. It's called Xenopus laevis. And he's got a Nobel Prize for this discovery I'm about to tell you. And, I mean, it's pretty obvious. He's also got some sick fucking hair, bro. I mean, look at that. Look at that mop. Look at them sultry eyes in that mop. John Gurdon. What are you doing, brother? There was a very common kind of assumption at the time, or a hypothesis. So if we take a stem cell, so we got a stem cell, an embryo. This is of the African clawed frog, also known as Xenopus laevis. Very cool looking frog, pretty goofy. And eventually this thing, like we said, differentiates is what it's called. It divides and it becomes all these other cell types. Lots of cell division and differentiation, which means it becomes specialized. Differentiation means becomes specialized. And that will, for example, then make, I don't know, let's say they become intestine cells. And intestine cells got a very specific shape, function, and all that jazz. So they got this columnar shape. They've got nuclei. And then if you remember, they got these little microvilli on them to help absorb, to create larger surface area, to absorb nutrients in your small intestine, for example. Right? We've gone from a just big, giant circular embryo into now we're a very specialized cell for a very specialized job. We've differentiated. So these are now an intestine cell. And a major question at the time was, how does this occur? What's going on? How did this work? Well, this cell must contain the DNA to become any cell. So how does a cell then become an intestine cell? Well, originally they thought something like this happened. Got ourselves a little trash can. And they thought an incorrect popular hypothesis, or kind of idea, hypothesis, was that the stem cell threw away instructions to become other cells. I can really not type today. Okay. John Gurdon showed that this is not true. Showed this isn't true. Okay. And how did he show this? That's what we're going to talk about. Okay. So what John Gurdon showed is that intestine cells still contain all the instructions for DNA, genes, to become any cell. And he did this by trying really hard, honestly, to find an organism that this worked in. And that was the African clawed frog. All right, so let's walk through this experiment. It's a beautiful experiment. Okay. So what he did is he took a little, not a fully adult frog, but he took a tadpole. Something like that. Got a little eyeball there. And tadpoles they got some guts, right? They got an intestine. All right, so this is a tadpole of an African clawed frog, tadpole. And what he did was he took some intestine cells, took them out. I'll draw them in this color. Swapping the colors here. And they got their little microphyllae. All right. So I took the intestine cell. I'm going to draw another one of these, like so. Okay, and he took an egg. It's not been fertilized. It's an unfertilized egg. Unfertilized egg. Right, so this would be actually no sperm has come into contact with it. It would technically be haploid, but technically actually wouldn't be haploid if you remember. But I'm getting a little off track here. Unfertilized egg. Okay. So this would be a stem cell as soon as it would be fertilized. And what he did was he took out the nucleus. Okay. It's called denucleated it, or took out the nucleus. And then what he did was, you guessed it, he put this nucleus in here. So he took this nucleus out here and put it in there. Okay? Put in the nucleus of an intestine cell. Okay, pretty brilliant little experiment here. Okay, and then he did some Frankenstein stuff. Boom. To kind of jostle this into thinking it's now fertilized, to get it to divide. And what do you think happened? He took this intestine nucleus out of an intestine cell, put it in the egg that's been denucleated, and then kind of tricked it to kind of think it's now ready to divide. And lo and behold, bingo. he got an adult frog. It developed into an adult frog. Pretty crazy stuff. Obviously, that is a beautiful froggy. He needed some upper hands too. There we go. Yeah, he's a little keyboardist. He's a typer. Yeah, maybe he needs a little frog nose. Like that. Boom. What does this actually say? This says that an intestine cell contains the instructions for all other cells, right? This frog got a brain, he's got a heart, he's got muscles, he's got skin. Took that, boom, it's now an adult. Right? So he took this old idea that the way a cell became specialized was to become a nerve cell, it would throw away muscle cell genes. To become a muscle cell, it would throw away bone cell genes. To become an intestine cell, it would throw away stomach genes. But that's not what they're doing. They're just turning different genes actually on or off, but they still have all of the genes. Okay? All right, so now let's... Oh, in this process here, what is this? What did we actually just do? We took this frog here, and we cloned it. This is cloning. This is also cloning. And we've done this with other animals too. The very first mammal we did it with was a sheep called Dolly. And it had problems. It lived to be about 6. It had problems with its telomeres. We'll talk about what those are. That's why I only lived to be 6. Okay, pretty crazy. Ugh. You see why they got the Nobel Prize for that. Thank you, John Gurdon, with your sick fucking hair. All right, now let's fast forward. And also got the Nobel Prize for this. Let's pull up another gentleman here. This here is Shinya. Hair maybe not so amazing, but findings just as incredible. Okay? Shinya Yamanaka. Shinya Yamanaka. Freaking maybe an even crazier experiment. Okay, and this was happening not that long ago. Maybe 2010, 2010s, maybe 2008, something like that. Somewhere around there. Not that long ago. Recent. Maybe some of you were alive. I certainly was. Okay. Got ourselves a stem cell. Shinya wondered, how do we take a cell like this? Let's say it's a specialized Crawley cell. Maybe getting a little confusing here. He took a fibroblast, which is a very specific job of laying down your extracellular matrix, collagen fibers, reticular fibers, etc. So we have a specialized cell, differentiated. It's called a fibroblast. Fibroblast. It lays down your extracellular matrix and your connective tissues. If you ever have a cut and then you get a scar, you could think you're fibroblasts. They laid down the reticular fibers to make that scar. All right. Shinya had the crazy idea to ask this question. Can we turn a differentiated cell into a stem cell. Right? Obviously, we can make stem cells. Stem cells become these, but can we turn them back there? This is normally what happens. As it normally goes, stem cells become differentiated. But can we turn a differentiated cell back into a stem cell? All right, so that's the question. Then I'm just gonna, we'll redraw. It won't take that long. I'm gonna redraw that differentiated fibroblast. I love how they look like little ghosts. Little sideways ghosts. Okay, and he was successful. He figured this out. He figured out how to do it. Boom. And he turned it back into a stem cell. It's not an omnipotent stem cell. It's what we call a pluripotent stem cell. Pluripotent stem cell. Meaning many. Powerful. Can become lots of other cell types. Not all of them, but lots. Okay? That's what that means, pluri. All right, so we go from a fibroblast or a specialized differentiated into this. And how many genes do you think he had to change, to turn on in order to do this? 1,000? 100? 50? 20? 10? It was 4. There's 4 genes. Changed 4 genes. Only 4 of them. You have like 20,000. You have to change 4 genes in a specialized cell to turn it back into a pluripotent stem cell. That's pretty freaking bonkers. And guess what? All of these are associated with cancer. All right, and this finding has huge impacts on health. For example, it's important for regenerative medicine. So for example, we could take one of your skin cells or whatever kind of cell we want, turn it into a pluripotent stem cell, put that in a dish, regrow you a new muscle, regrow you a new heart. That's the goal. Like, that's what we're working on. Regrow you an entire new lung, new kidney. Like, that's the goal. Could do things like increase our lifespan, right? If we could look at your cells genetically and be like, oh yeah, you got some of your aging genes turned on. Here's some stem cells to kind of replenish some of your tissue. We could also cure various diseases. Like, oh, your bone marrow cells aren't making you enough red blood cells. Let's take some of your skin cells, turn them into more bone marrow cells. Boom. Put some more of those in there. Now you got enough. Now you're making enough red blood cells. Okay. And it also just shows you how related cancer is to being a stem cell. Like, if you think about that, we all started off as a cancer. right? Because cancer cells are more stem-like. Okay, and the next one we're going to talk specifically about cancer. That's all I want to talk about with stem cells. It's some pretty mind-blowing stuff there. Shinya Yamanaka also got a Nobel Prize for this. They got it together. John Gurdon had to wait a long time for his Nobel Prize. He did that work in the 60s, didn't get the Nobel Prize till Shinya Yamanaka did his work. So, sometimes you got to wait for your recognition. All right. We'll talk about cancer in the next one. See ya.